Hi, and welcome to the Radiation Research Society vodcast. I'm Allison Burrell, and I'm here interviewing Gayle Woloschak. Welcome. Thanks for coming to talk with us today.
Happy to be here.
Thanks. Now, you are the departing president of the society.
Yes, thankfully. So it's good news.
Yeah, so I'm just curious what goes into being president of this society.
You know, it differs from year to year. This past year was complicated. I mean, the year when you say you're going to be president of the society, it's a 4-year commitment. 1 year as second vice president, 1 year as first vice president, 1 year as president, and 1 year as past president. So during the first vice president year, you run the meeting, and during the president year, you run the society. So it's handling all the interactions from one group to another, forging better relations from one society to another. So for instance, we have relationships with National Council on Radiation Protection, with Health Physics Society, things like that. This year was especially interesting, shall I say, because of the International Congress of Radiation Research in Kyoto.
Right.
So that was a very big part of my time, was trying to get us well placed within the program, interface with them, get our membership to be able to attend that meeting. But it does vary from year to year depending on what the ongoing activities are.
And that was a very exciting meeting.
It was really a great meeting. I mean, the Japanese, there was a lot of trouble getting things set up, and I won't go into the details about it. I'm not sure that's relevant for anybody, but the Japanese put forth a really big effort to put on a great show, great program. They tried to be very hospitable. Had some wonderful events that were just incredible. I mean, for me, Leah, I can't remember Leah's last name, but she's Al Fornace's person, works with her. She did a video of me breaking the sake cask.
Oh, yes.
It was so cool. I mean, and that was really fun.
I guess you had to think about this 4 years ago, then becoming the president. Was it something that you had considered, or?
Oh, it was like, it was totally random. So I never thought about being president of Radiation Research Society ever, okay? And Frank Cucinato, who was the president before me, he sends me an email and says, "Oh, look, Gayle, we don't have many people to run for president. Would you be willing to do it?" And I was like, "Oh, sure, what the heck, you know, I'll happy to help, okay?" And next thing you know, there are 4 people running. All four. I would not have run against any of the other 3. They all would have made great presidents. But something happened. I won, and no. But the really great thing is that the 3 people who didn't win were remarkably supportive, and that's the wonderful thing about the society, is that everybody's in it for the good of the organization, and they were really great about that. Society's been really supportive for everything I've wanted to do, so it's really been a good thing.
Has it been hard to balance working, running the society and with all of the work that you do in your own lab?
Well, so what I would say is, first of all, my chairman is extremely supportive. When I was running for office, I told him about it, and he said, and he's a member actually, and he said, "Oh, look, Gayle, I'm going to support you. We want you to do this. We'll do whatever we can." So he's at least given me moral support and doesn't seem to mind when I have to miss a few lectures for the residents because I have to travel for rad-res or things like that. I think it's just the support system that helps.
So this past year you've had to travel a lot to...
We've had a few more meetings because of it, certainly a lot more conference calls, a lot more emails, a lot more letters. There's a lot of paperwork time that goes into it too. But again, great support system. I mean, Veronica and A Meeting by Design are really helpful. Ellie Blakely is the secretary treasurer. She does most of the heavy hitting on the correspondence. So there are a lot of people there to help out.
Yeah, I spoke with her in Japan actually on her work.
Is that right?
It's on a podcast as well.
Cool, cool. She's great.
Yeah, but I guess diving into your work, I know that you've got a couple of things going on. One is the repository of samples. You want to talk a little bit about that first?
Yeah, so that's sort of an interesting thing. I mean, we've had, the U.S. Department of Energy funded, back in the 1960s, 70s, 80s, experiments with 49,000 mice and 21,000 dogs that had been irradiated under different conditions, different circumstances, different dose rates, different qualities of radiation. And the story goes, I was moving from Argonne National Lab, where I had at least the 49,000 mice with me. And I said, look, you guys, I'm leaving, can I take the tissues with me? And they said, oh, no, we're going to work with those tissues here. So I said, okay. I went to Northwestern, never thought about it. 2 years later, somebody at Argonne calls me up and says, Gayle, the tissues are in the dumpster. So I called Department of Energy. I said, there's a problem. All these tissues that I told you I really could use, they're in the dumpster right now. So DOE said, we'll give you a grant, we'll move them down to Northwestern. So now I had all these tissues I'd worked with. I think even more importantly, we have the data sets. And in the day and age of big data, there's so much you can do with large data sets that it's extremely useful. So then what happened was people heard, investigators are passing away. They worked on these back in the 60s. What are we going to do with the tissue? So now everybody's sending me their archives. So I've got.
So it's getting bigger.
It's getting bigger and bigger, and now I have the dogs from Lovelace and some rats from Lovelace, and P&L sent me their dogs, and so it accumulates. But what else is really cool is when we're doing this, the EU is doing a similar project that they called STORE, where they're storing tissues, and they're developing a website. So now we combined our websites. You can actually access our data from their website and vice versa. Their data on 250,000 mice that have been irradiated, just mice alone, in the archive, in the worldwide archive. EU-US archive. We're trying to get Japan to enter into it. I mean, if people keep adding their data, we can have a huge data set of studies.
And by data, you mean expression profiles?
Some of them, people can upload expression profiles. Back in the 60s, 70s, 80s, they didn't have that sort of information. What they had was, you irradiate the animal, what was the micropathology, what did you see in every single tissue, and we have tissue blocks to go back to. So, for instance, we've done a number of studies where we've been looking at microRNAs in the irradiated samples. We've identified microRNAs that are important, and then we go back to new animals to now look at the experimental systems that we can modify a bit. So we go back and forth between archive and fresh samples. But 49,000 mice, I mean, nobody's going to do an experiment like that again.
No, that's...
So it's a wealth of information. It is mostly paraffin. For the dogs, we have some leukemia samples too. So. Okay.
And then the other topics that you're working on in your lab are utilizing nanoparticles?
Yeah, so we have a nanotechnology project. About 10 or 15 years ago, when I was still at Argonne, some chemists came to us and said, hey, we've got these nanoparticles. What would you biologists think about putting on the surface? And we said, well, the easiest thing is to put DNA on the surface. So we started working with them collaboratively. And then as work progressed, it turned out that we were working with titanium dioxide, which actually has radiosensitization capacity. When you irradiate it, it gives off free radicals. And so now if you can put those nanoparticles close to the DNA, you can actually get them to radiosensitize. And that's what many of my students, we've published a few papers showing that radiation alone, gives gamma H2AX, P53, 53 BP1, at high levels. But now put the nanoparticles in and it goes way high. So we can get better killing, we can get better DNA strand breaks. If we can make it be tumor specific, which we can do by modifying the surface to target a tumor antigen, we can create radiosensitization.
Those studies, are those just in cell lines right now, or have you moved into animal models?
We've been doing a little bit in animals. We've done a little bit with mice, but probably the more important model for us is the rabbit. The reason why is because we're actually using a patient irradiator. So we're trying to, so little do the patients know, but at night there are rabbits being irradiated. We have to really clean it down when we're done. But we put the rabbits in, and we actually do a treatment plan with a CT scan, just like you would do for a patient. And then we irradiate the tumor with IMRT, and look to see whether we're getting better killing with nanoparticles or without. The jury's still out. We have a few experiments that look very promising, but we've larger numbers to do and stuff. So,
are there any other projects that you're?
Those are probably the two most important. We have a third project that really dovetails with the others, where we do X-ray fluorescence microscopy, which is a synchrotron-based form of microscopy, and we developed the BioNanoprobe, which is an instrument at Argonne synchrotron. And it allows us to detect things like plutonium in cells. So we're relating that back to the dog studies where they gave animals plutonium, and at the time the only way to track plutonium was with radiolabel. Radiolabel isn't such a good way, it's not very sensitive. So now we can look at tissues and actually tell you how much plutonium there is where, and what's the oxidation state of the plutonium. So it's pretty amazing.
That does sound
And this technique didn't even exist 10 years ago. So
there's new technology coming out all the time.
Yep. I mean, everybody thinks about the molecular technology, but microscopy is also, really developing into new approaches. And synchrotron-based microscopy, I think, is going to be a wave of the future.
So I know you had spoken a bit about being president and being president of the Radiation Research Society, that you didn't know if you were, back when you were first starting out, that you were going to do radiation research. So I'm just curious if you have any ideas of where your research is headed. I know you start with one thing and it leads in some direction and it's completely off of what you were originally interested in. Do you have any aspirations for new projects?
Yeah, well, we have a couple of directions that have been, for me, very important. One of my students presented a poster here on DDREF, which is the dose and dose rate effectiveness factor. And he was using large numbers of animals to the 210, 250,000 mice. Where I see next directions going are to try to do much more big data work in radiation research in general. So what it looks like to me is that there are some pretty good databases out there that people have developed here. I think we need to come up with a way to somehow link those so that it's much easier to access the information. You can certainly get information on patients that have been irradiated and get gene expression profiles and stuff like that, but I don't think you can get it in one place anymore. So it exists out there, but it's not in one place. I think we need to work as a community to do that so that we can now start to mine those data in a new way.
So definitely bioinformatics.
I think so, and especially for us with the low-dose work that we've been doing and the large-scale animal studies, that is about data mining almost exclusively. But if we can now start to put together many more things, I think we'll be much better off. I tried to say this in the President's Symposium yesterday, but I think the link that's missing in radiation research right now is we do the omics, we do the molecular biology, we do the metabolomics, we have all that work. And then we have the epidemiology where we look at people and try to do cancer risk assessment. But translating from one to the other is very, very hard. And it's up to us, actually, to come up with tools to be able to bridge that gap. National Council on Radiation Protection and Measurements, they've done a little bit of work trying to say, oh, there are biomarkers that might be useful, but we need somehow to bridge that, and I think that's going to take a lot of work.
Right, because the symposium was on low-dose radiation.
Yeah, it was on low dose, which is where I think there are big challenges.
Right.
It's much easier to understand what's happening at high doses because there are a plethora of responses. A lot of times low dose, you only know by looking at high dose and extrapolating down. When you're looking at whole populations, there's so many confounding factors, it's really hard to do that. But yet, in a way, that's what we're supposed to be doing. There's this comics, I mean, it's not really a joke, but people told it as a joke at the time, where they're women who are pregnant at Chernobyl, and they're asking the radiation biologist, what should I be doing for what's my baby's risk from? And the radiobiologists are off on the side talking about how cancer is induced by radiation and oncogenes and stuff like that. So we're not really answering the question that is in people's minds. I think we haven't yet developed good tools to do that, but that's the challenge we have to do.
Okay, great. Well, thank you so much for coming and talking to us.
My pleasure. Good luck. I hope everything keeps going well.
Thanks, Gayle.